This is all about assessing and stabilizing neuroplasticity in autism spectrum disorder using transcranial magnetic stimulation. So autism spectrum disorder is the most common neurodevelopmental disorder, which approximately affects 1% of Canadians. Autism spectrum disorder basically have two core symptom domains. One, there is a pervasive deficit in social interaction and verbal and non-verbal communication. And also, two, there is presence of highly restricted and circumscribed interests and repetitive behaviors. When we make a diagnosis of autism, both these symptoms should be present. Interestingly, it is true that the number of cases of autism are on the rise. In fact, there has been over 40-fold increase in the cases of autism over the past 4 decades. And the most recent estimate indicates that probably 1 in 68 children suffer from autism, so which makes it the most common neurodevelopmental disorder, even more common than the common major mental illness that we know, such as schizophrenia or bipolar illness. So yet, the cause of autism is still unknown, and we do not have any identified cure, even after 7 decades of research. So really, there is time to think out of the box. So one mechanism that has recently gained a wide attention implicating its causative role in autism is abnormal neuroplasticity. So what is neuroplasticity? Neuroplasticity is basically a fundamental property of our brain. When the brain constantly alters and reorganizes its connections in response to both internal and external stimuli. So our brain is very dynamic. It's constantly trying to adapt to modifying stimuli. Perhaps one simple example of neuroplasticity would be memory. Like, we tend to retain facts even after we finish reading the book. We tend to retain scenes even after we've finished watching a movie. So the most important property of neuroplasticity is that brain modifies itself to retain information, and which is retained even after the stimulus that caused it ceases to exist. So brain tends to retain this information over a period of time. So this is a fundamental property of neuroplasticity. So the regulation of neuroplasticity, it involves a very delicate balance between facilitation or excitation and inhibition. So we have 2 types of neuroplasticity. This is important to understand my research, actually. So one is long-term potentiation, like we call it LTP, and other one is long-term depression, which is LTD. We all know that practice makes us perfect. This is an example of long-term potentiation. This is achieved through long-term potentiation when the brain connections get stronger and stronger. The other example is long-term depression. Perhaps a simple example of long-term depression would be like most of us, or maybe some of us, like at some point of time, we all experience some bad habits that we want to unlearn. So the way we unlearn this bad habit is by long-term depression. So that is one example. So I will not go into the 2 details, but so the fact that brain in autism is characterized by abnormal neuroplasticity is supported by 4 key lines of research. The first thing, we have mutations in genes, several genes in fact, that actually regulate our brain plasticity. The next line came from neuroimaging, like magnetic resonance imaging and computerized tomography scans. But I will highlight the 3rd and 4th, which is quite relevant to my research. Animal experiments. Now, animal experiments give us a direction of neuroplasticity abnormality. So when we're talking about abnormalities, there can be two types of abnormalities. One, either neuroplasticity is completely deficient. We call it deficient neuroplasticity or hypoplasticity. The other example would be when the neuroplasticity is abnormally excessive. We call it hyperplasticity. There can be only two types of abnormalities. The interesting thing is that we can manipulate mice and produce autistic symptoms in mice. We do it by either manipulating the genes, or we administer medications like valproic acid, and we induce autistic symptoms. It has been found that mice who display typical autistic symptoms, they have an abnormally, the brain is characterized by abnormal hyperplasticity, like excessive neuroplasticity. So this is perhaps one biological basis to understand autistic phenotypes, autistic behaviors. Like if the brain is hyperplastic, like if their brain is characterized by too much plasticity, we'll talk about more subsequently about it. Then you can suffer from autistic symptoms. So what would be the example of, like, how would hyperplasticity manifest itself clinically? What do you see? We do not have the answer yet, but I can give some examples which could be related to hyperplasticity. Now, we know that people who suffer from autism, sometimes they do not feel any pain. We had seen clients like, who literally eat up their flesh and feel no pain. Or on the other hand, you can see clients like who cannot tolerate even wearing a shirt or like particular fabric, or who do not need to be cuddled. They hate cuddling. They cannot tolerate simple touch. So either they are excessively sensitive to touch, or they can be like extremely tolerant. This could be one example of like excessive neuroplasticity in the brain. So the fourth one that is quite relevant to this project is transcranial magnetic stimulation. This involves human subjects. So this kind of gives us a direct evidence of abnormal plasticity in the brain. So I'll try to explain in simple terms what transcranial magnetic stimulation is. Basically, we have a figure of 8 coil, and when electricity is transmitted through it, it creates a magnetic field according to Faraday's principle. So that magnetic field, when we hold the coil on particular area of the brain that we want to stimulate or inhibit, then the magnetic pulse that is generated that either stimulates or inhibits, depending on the properties we're using, the neurons that are underneath. So this is a very innovative way. We are not actually directly applying electrical currents, but rather we are using the magnetic field to generate electrical current inside the brain. So the advantage of transcranial magnetic stimulation is that it has a sophisticated ability to really focus on target areas of the brain. Now, if you consider drugs, like if you take a medication that is distributed all over your brain, so the area that does not need to receive the molecule is also receiving the molecule. So that adds to the side effect. So here we are bypassing the side effect by just focusing on the area that we call it region of interest, and we just focus on that. So transcranial magnetic stimulation is very effective. It is very well tolerated. It is safe. Okay, so the type of magnetic stimulation I'm using in my study is called theta burst stimulation. Now, I'm using this theta burst stimulation to assess neuroplasticity. Now, how do we use magnetic stimulation to assess neuroplasticity? We cannot see neuroplasticity because neuroplasticity is happening inside your brain. But we have a way to measure it. We measure it by tracking thumb movements. We stick electrodes on thumb, and then we measure the thumb movement potential. We call it motor evoked potential, or MEPs. Now it is very simple. If we stimulate the area of the brain that regulates the motor cortex, we call it the area of the brain which regulates thumb movement is called the motor cortex. If we stimulate the motor cortex, that will facilitate thumb movement, very simple. If we inhibit the motor cortex, it will inhibit the thumb movement. Now, theta burst stimulation, there are two types of theta burst stimulation. One involves intermittent delivery of pulses, magnetic pulses, we call it ITBS. And the other one is a continuous delivery of magnetic pulse, we call it CTBS. ITBS stimulates the motor cortex, so it facilitates thumb movement. CTBS inhibits the motor cortex, so it inhibits thumb movement. Now, what happens when we deliver theta burst stimulation, it is delivered within less than a minute, like 40 seconds. Interestingly, after we deliver ITBS to motor cortex, which facilitates thumb movement, when we measure this thumb movement, we see that thumb movement is facilitated for half an hour. And when we deliver CTBS, which inhibits the thumb movement, when we continue to track thumb movement, the thumb movement, inhibition of thumb movement is facilitated again for half an hour. So remember the rule of neuroplasticity. Brain is retaining the response even after the stimulus that caused it ceased to exist. So we stimulate the brain for 40 seconds or inhibit the brain for 40 seconds, and thumb movement, either suppression or facilitation, is maintained for half an hour. So this is a way to study neuroplasticity. So the next slide will elaborate. Now, if you look into the graph, so the blue lines and the red lines, so the duration of facilitation of thumb movement is what is of our interest. So you see, like, the blue lines is coming back to baseline in 40 minutes when we are applying ITBS, and the red lines are coming back to the baseline around 25 minutes when you are applying CTBS. So this duration of suppression to CTBS is an index of long-term depression. And the duration of facilitation of thumb movement to ITBS is an index of long-term potentiation. I hope it's making sense. So this is a very innovative way to study neuroplasticity in the brain. Now, so this experiment was done in Harvard lab, in Harvard University, and Lindsay Oberman is a scientist that I collaborate with her. So she's the only researcher in the world who actually applied this method on adults with autism. Now I will explain what is happening here. The red lines represent ASD, and the blue, now purple, it is almost black now, represent controls. So if you focus on the red lines, now top is ITBS, and at the bottom half you have CTBS. You can see that it is very obvious that the blue lines are coming back to baseline in around 40 minutes, and the red lines are coming back to baseline in around 100 minutes. So it is taking way long for the facilitation or suppression of thumb movement to come back to baseline in case of people with autism compared to controls. So what does it tell us? It tells us that the brain in autism is way too plastic if we compare this with controls. So again, the studies, TMS studies, is basically indicating that brain in autism is characterized by excessive neuroplasticity, which is kind of consistent with what animal experiments were telling us. So now this is a direct evidence that the brain is hyperplastic. So now so with this background, I just share the goal of my research project. So basically, we like to promote a new line of therapeutic intervention in autism, which is based on the core pathogenic mechanism of autism. I have two objectives. So the first objective is using this ITBS and CTBS. I want to basically confirm that the brain in autism is indeed hyperplastic. And there is a second objective, where I'm applying another RTMS, like we call RTMS means repetitive transcranial magnetic stimulation, another RTMS, to see if we can reverse, if we can stabilize the excessive hyperplasticity in autism. What is the purpose? Because I already shared that key lines of, like 4 key lines of evidence indicating that brain in autism is actually characterized by hyperplasticity, which could be directly related to the symptoms of autism. So what will happen if we directly modify the core biomarker of autism? We expect that, that it will lead to very robust improvement, probably cure, of autism. So in this regard, stabilization means that we want that neuroplasticity measures to be comparable with controls, healthy controls. That is what we are trying to see with this RTMS intervention. So the next question is, so if a child is born with hyperplasticity, and now the child is an adult, so why are you telling us that by applying magnetic stimulation you should be able to rectify and completely reverse a deficit that has been present for a long time? So there recently a very interesting paper has been published in Nature, which is like top journal in science. So here researchers, what they did, they turned off a particular gene, we call it SHANK3, in a pool of mice, and the mice experienced typical autistic symptoms. They allowed the mice to grow into adulthood. And then they turned on the gene that they previously had switched off. And what they noticed is that completely reversed the autistic symptoms, even in adult mice. So this is a very recent paper which was published in Nature. So this is first direct evidence that it may not be impossible to reverse a fundamental pathology of brain in adulthood. So this gives us hope that it is possible, of course. So in this, we're talking about what we need is an optimal amount of plasticity. So for our performance, for our cognition, for brain health, we need optimal level of plasticity. If the brain is less plastic, then the cognition and performance will suffer. And if the brain is way too plastic, it is not good either, because that will lead to a chaotic brain connections. And perhaps this is the reason why like 3 to 4 people out of 10 who have autism, they experience epilepsy. Now, epilepsy is a signature of very chaotic brain structure. So that could be one of the reasons why we see more cases of epilepsy in people with autism. This could be, again, due to this hyperplasticity. So basically, we need an optimal amount. So I apologize for the very busy slide. I will simplify. You don't have to look, those who want to see the details, it's there. So this basically talks about why are we claiming that this particular RTMS intervention will stabilize neuroplasticity. So basically, we are using 2 types of stimulation, the theta burst stimulation to assess neuroplasticity, and we are using another magnetic stimulation to reverse neuroplasticity. Now, in this slide, I'll be talking about the rationale behind using the RTMS to stabilize neuroplasticity. Now, in simple terms, regulation of neuroplasticity involves a delicate balance between excitation and inhibition. In autism, we know that excitation prevails over inhibition. That is known neurophysiologically. So this RTMS intervention is supposed to boost underlying inhibitory neurotransmission. We hope that if we boost inhibitory neurotransmission, then the excitation-inhibition balance will be restored. If this is restored, we expect that plasticity abnormalities will disappear. So the particular RTMS intervention we are using in the lab, we found that compared to all other RTMS procedures, this particular RTMS procedure has the most robust impact on enhancing inhibitory neurotransmission in the brain. So that is the reason we are using it. So we want to, restore the delicate balance between excitation and inhibition. These are my subjects. We are recruiting high functioning. High functioning means like those who do not have any intellectual disability. This is because like they tolerate the procedure better, and you can see like we are recruiting people who are currently stable, and yeah. So this research is being taken place at Temarty Center for Therapeutic Brain Intervention at CAMH. So this is a state-of-the-art facility to conduct this brain stimulation research and also neurophysiology research. So this is the design. So you can see there are two phases. First phase, we are assessing neuroplasticity so that everybody, like people who have autism and healthy controls, everyone will receive both types of theta burst stimulation I talked about, like ITBS and CTBS. This is the assessment phase where we are assessing, and then we are trying. And then there is a second phase, we call the intervention phase. Only people who have autism will receive that intervention, because for healthy controls, we have nothing to stabilize, they are healthy. They will receive active intervention, and of course, that's a gold standard in medicine. We always have to compare this with placebo or sham. So active has to beat sham, otherwise we don't say that it's effective. So they will receive active and sham, and they'll come back on the next day where we will again test for neuroplasticity to see like what change has taken place. What is happening there? Has it been reversed or nothing has happened? So for that reason, they are coming later. So I will share a couple of slides. This is really preliminary data. So this data is for 8 adults with autism and 6 healthy controls. Again, the red lines represent autism and the blue lines represent healthy controls. You can again see even with 8 subjects, you can see if you concentrate on the top red line, it takes long time, for the facilitation of thumb movement, which is the index of long-term potentiation, to come back to baseline, almost 120 minutes, 2 hours. And if you compare the blue line, it is coming back to baseline at around 35 minutes. So it's way prolonged. And similar thing is happening in case of CTBS as well. So already with 8 subjects, we are getting an indication that brain is hyperplastic in autism. So this is one example of whether or not the active RTMS is really stabilizing something. This is again a preliminary data. In this data, you can, so the blue line is the pre-active RTMS neuroplasticity, and the red line represent like it's a post-active RTMS neuroplasticity. And you can clearly see that active RTMS has attenuated the plasticity to baseline level, comparable level. So this gives us hope that probably this intervention might work, but this is really preliminary data. We don't theorize with 8 subjects. We have to wait and see whether we see similar trend as we continue our experiment. But this data is really quite promising so far. So what is the significance? As I told, after 7 decades of research, we have no cure of this condition. And the number of cases are on the rise. There are over 70,000 people who suffer from autism. There are 70,000 adults in Ontario who suffer from autism, and they have no services. They don't have any treatment. So this is the real innovation of this project lies in the fact that we are trying to reverse a fundamental pathology in autism. So this has never been tried before. So that's why I call it a proof of principle, proof of concept study, where it's a theory that we need to test that if we can rectify this abnormality. if we really can do, if we are successful, then what will happen? Well, we hope that if we are successful, then it will lead to very robust improvement in autism. As I shared, like the mouse model, like when they turned on the gene and then the symptoms disappeared. That is like magical, but we don't expect magic with the human brain. But so far the data is quite that promising, I would say, and also like this will also expand our understanding of neuroplasticity in general. Like we can manipulate neuroplasticity. If we can stabilize neuroplasticity, then obviously like we'll have more control over brain function. And also, if you are successful in adults, then the same method can be applied in children, because we need to see the safety data, and this can be done in children. So this is my team of investigators, and yeah, so I think that's it. So I again thank the foundation for really supporting this line of inquiry, which we scientists believe like very, very important. Yeah, thank you so much.